Welcome to the PubMed Daily Review for April 25th, 2026. Today we scanned 2,917 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5. Gene editing for disease treatment. These papers explore biological mechanisms and therapeutic interventions for various diseases, with a focus on genetic and cellular approaches. This cluster covers 797 papers. Representative work includes in vivo adenine base editing of mutant Galc gene ameliorates Krabbe disease progression, investigation of the clinical efficacy of mesenchymal stem cells in combination with monoclonal antibody therapy against feline panleukopenia, and retraction. Effect and mechanism of inhibition of PI3K/ACK/mTOR signal pathway on chronic neuropathic pain and spinal microglia in a rat model of chronic constriction injury. Trend 2 of 5. Predicting health risks with data. These papers unify in using data-driven methods to predict health risks and outcomes across various medical conditions. This cluster covers 642 papers. Representative work includes risk prediction of combined fibrinogen degradation products, thrombomodulin, and maximum amplitude for assessing association with venous thromboembolism in patients with post-traumatic fractures, quantification of pulmonary arterial pressure with 4D flow cardiac MRI velocity mapping in patients with suspected pulmonary hypertension, comparison with right heart catheterization, and editorial note, risk factors for mild depression in older women with overactive bladder syndrome, a cross-sectional study. Trend 3 of 5. Material science and water chemistry. This trend explores how material properties and chemical interactions, particularly with water, drive diverse applications. This cluster covers 402 papers. Representative work includes a density functional theory-based investigation of a pH and redox-driven treatable 2-rotaxane and CH2 dilute solution, plasma-activated water, mechanism and treatment duration for post-harvest disease control and shelf life enhancement of mango under ambient storage, and the microscopic mechanism of water immersion and collapsibility in malonlus with different particle size. Trend 4 of 5. Cancer progression and signaling pathways. These papers investigate molecular mechanisms driving cancer growth, metastasis, and recurrence. This cluster covers 407 papers. Representative work includes retraction. Loss of FOXN3 in colon cancer activates beta-catenin/TCF signaling and promotes the growth and migration of cancer cells. Retraction. Fibulin. 4 is associated with prognosis of endometrial cancer patients and inhibits cancer cell invasion and metastasis via WNT/beta-catenin signaling pathway and retraction. By downregulating TIM1 expression, microRNA 329 suppresses gastric cancer invasion and growth. Trend 5 of 5. Healthcare practices and patient outcomes. These papers explore how healthcare practices influence patient health and well-being across diverse contexts. This cluster covers 669 papers. Representative work includes comparative risk of psychiatric comorbidities associated with codeine and tramadol in patients with hip osteoarthritis, a nationwide population-based cohort study. Time and target range for body mass index and risk of new onset multimorbidity in middle-aged and older adults, a landmark analysis of 2 prospective cohorts, and enhancing health professional clinical performance through supervision strategies from 5311 facilities across 8 countries in sick child healthcare. Up next, our first paper out of 5, Clinical Development of Molecular Residual Disease, MRD, and Multi-Cancer Early Detection, MCED. Using liquid biopsy multiomics with artificial intelligence, AI, by Taro Shibuki and Takayuki Yoshino from Hepatobiliary and Pancreatic Oncology, National Cancer Center Hospital East, Kashiwa, Japan. Let's break down its key contributions. Cancer remains a leading cause of death worldwide, and early detection is key to improving patient outcomes. Unfortunately, current screening programs only cover a limited number of cancer types. This leaves a significant gap in our ability to catch many cancers early. Furthermore, monitoring for cancer recurrence after treatment, known as molecular residual disease, is also challenging. This study aims to tackle these issues by developing more sensitive and specific tests using advanced technologies. This study employs a comprehensive approach, leveraging data from 2 large Japanese cohorts, Monster Screen 3 for cancer patients, and the TMMO project for cancer-naive individuals. They are integrating a wide array of omics data, including genomics, transcriptomics, proteomics, metabolomics, and microbiome profiles. This vast dataset is then fed into sophisticated artificial intelligence models to develop advanced classifiers for detecting cancer and pinpointing its origin. The goal is to create highly sensitive and specific assays for both early cancer detection and monitoring residual disease. While this abstract focuses on the development phase, the projected performance metrics are very encouraging. The study plans to analyze data from approximately 1,000 cancer patients and 1,000 cancer-naive individuals to build their AI models. Based on these numbers, they anticipate a strong performance for their cancer detection classifier, with an area under the curve, or AUC, ranging from 0.83 to 0.97. Similarly, the classifier for predicting the cancer's origin is also expected to perform well. These results will then be validated in smaller, separate groups. This study represents a significant step forward in the field of cancer diagnostics. By integrating multiple layers of omics data with artificial intelligence, it aims to overcome the current limitations in sensitivity for early cancer detection and MRD monitoring. The ultimate goal is to develop more accurate and accessible liquid biopsy tests. If successful, this approach could revolutionize precision oncology, leading to earlier diagnoses, improved treatment strategies, and better patient outcomes. Up next, our second paper out of 5, From Prompt to Drug, Toward Pharmaceutical Superintelligence by Alex Zavaronkov and G.S. Sherfrom. 612614 in Silico, Medicine US Inc., Cambridge, Massachusetts, 02138, United States. Let's break down its key contributions. The journey from a simple idea to a new drug is incredibly complex and time-consuming. But what if we could change that? Imagine typing a request and having an AI system design, synthesize, and validate a potential drug all on its own. This paper explores the exciting possibility of a fully automated prompt-to-drug pipeline driven by the latest in artificial intelligence and laboratory automation. This isn't just about AI designing molecules. It's about creating a fully integrated system. The prompt-to-drug pipeline leverages machine learning, deep learning, and large language models to not only generate hypotheses and design drug candidates, but also to control robotic synthesis and validation. Think of it as a digital scientist that can plan, execute, and learn, all within a closed loop. We're already seeing incredible results. For instance, the Gentral model identified and validated a DDR1 inhibitor in just 21 days and a CDK20 inhibitor in 30 days. Beyond discovery, systems like ChemAgents and SynBot are showing promise in automating the actual laboratory work, from synthesis to testing, paving the way for truly hands-off research. This research paints an exciting picture of the future, a drug discovery process that is dramatically accelerated and more efficient. While challenges remain, particularly in automating the synthesis of complex biologics, the convergence of AI and automation is undeniable. The next steps involve refining these systems to handle greater complexity and further integrating advanced reasoning capabilities to unlock the full potential of pharmaceutical superintelligence. Up next, our third paper out of 5, Single-Cell Mapping of Tumor-Driven Macrophage Reprogramming via ETV4, MC1, are underlies immunotherapy resistance in colorectal cancer by Rui Liang and Tao Wu from A Department of Colorectal Surgery, the third affiliated hospital of Kunming Medical University, Yunnan Cancer Hospital, Peking University Cancer Hospital Yunnan, Kunming, China. Let's break down its key contributions. Colorectal cancer, especially in its advanced stages, often becomes resistant to immunotherapy, posing a significant clinical hurdle. While immunotherapy has shown promise, a substantial portion of patients do not respond. We need to understand the intricate molecular changes within tumors that lead to this resistance to develop better treatments. To tackle this challenge, researchers employed a multifaceted approach. They started by analyzing tumor samples from patients who either responded or didn't respond to immunotherapy using single-cell RNA sequencing. This allowed them to build sophisticated models to identify key gene regulatory networks. These findings were then rigorously validated through a series of laboratory experiments, including cell-based assays and studies in mouse models. The study revealed that the transcription factor ETV4 plays a critical role in driving resistance by reprogramming macrophages. Mechanistically, ETV4 activates MC1R, leading to an immunosuppressive M2 macrophage phenotype. Crucially, in preclinical models, combining a therapy targeting the ETV4 MC1R axis with anti-PD-1 treatment resulted in a significant 55.6% reduction in tumor volume compared to anti-PD-1 therapy alone, with a p-value less than 0.05. This research uncovers a critical new pathway, ETV4, MC1R, that contributes to immunotherapy resistance in colorectal cancer by manipulating the tumor's immune environment. Targeting this axis alongside existing immunotherapies presents a promising strategy to improve patient outcomes. These findings pave the way for future clinical investigations to test this novel combination therapy in patients. Up next, our fourth paper out of 5, a transcriptomics-native foundation model for universal cell representation and virtual cell synthesis by Xiaowei Jiang and Jichuan Xie. Let's break down its key contributions. Analyzing cellular data, especially from transcriptomics, is crucial for understanding biology. However, existing foundation models often fall short because they don't fully embrace the unique nature of this data. This can lead to underperformance compared to specialized tools, hindering our progress in areas like identifying rare cell populations. We need a smarter way to represent and even generate cellular data. To address these limitations, we developed XVerse, a novel foundation model built specifically for transcriptomic data. It integrates batch invariant representation learning with probabilistic generation of expression profiles. This design allows it to preserve biological diversity while effectively reducing batch effects. The model also excels at synthesizing realistic virtual cells. The results are impressive. XVerse demonstrated a 17.9% improvement in representation learning and a 34.3% improvement in spatial imputation compared to leading methods. It can also generate virtual cells that are virtually indistinguishable from real biological data. Furthermore, XVerse enables accurate clustering and marker detection even in very small datasets, resolving rare cell types with as few as 4 cells. In conclusion, XVerse represents a significant leap forward in analyzing cellular data. Its transcriptomics-native approach unlocks new analytical capabilities, particularly in resolving rare cell types and improving data augmentation. This framework holds immense promise for advancing our understanding of complex biological systems and various disease states. We anticipate further developments and applications of this powerful tool. Up next, our fifth paper out of 5. In vivo adenine base editing of mutant Galc gene ameliorates Krabbe disease progression by Beigan Nam and Sung-Rae Cho from Research Institute of Rehabilitation Medicine, Yonsei University College of Medicine, Seoul, 03722 Republic of Korea. Let's break down its key contributions. Krabbe disease is a devastating inherited disorder that affects the nervous system, primarily due to a faulty gene called GLC. This leads to a breakdown in myelin, the protective sheath around nerves, causing severe neurological damage and typically a very short lifespan. Existing treatments have limitations, and there's a critical need for therapies that can fix the root cause of the disease. This study explores a novel gene editing technology as a potential solution. To investigate the potential of adenine base editing, researchers utilized the Twitcher mouse, a well-established model for Krabbe disease. They delivered a specific base editor, ABE8E, directly into the brain using a viral vector. This approach aimed to precisely correct the genetic error responsible for the disease. The study then meticulously assessed the editing efficiency, the restoration of enzyme function, and the subsequent impact on the disease's neurological and physical manifestations. The results were highly encouraging. The ABE8E treatment achieved approximately 5% correction at the mRNA level, which was sufficient to restore about 5% of normal GLC enzyme activity. This led to a substantial 47% reduction in the toxic cytosine metabolite. Crucially, treated mice showed remarkable improvements in motor coordination, with performance reaching 64% of wild-type levels, and experienced a significant extension in their lifespan, demonstrating the therapeutic potential of this approach. This study provides compelling evidence that adenine base editing can effectively treat Krabbe disease in a preclinical model. The ability to directly correct the genetic mutation, even at modest levels, resulted in significant functional recovery and improved survival. These findings pave the way for further development of base editing therapies, offering a beacon of hope for patients with Krabbe disease and potentially many other genetic conditions. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.